Welcome back, guys. This is lecture 13 of ZOO3649, Evolutionary Genetics 2020. I would like to give you this very special lecture, and we're continuing with the third lecture in sexual selection. And remember, like natural selection, um, there was evidence provided in a lecture of natural selection. Remember, we went through lots of evidence for natural selection, including the Grant studies of Rosemary and Peter Grant in the Galapagos Islands, and artificial selection is also evidence for natural selection. The peppered moths and so on. Lots of examples of natural selection, evidence for natural selection. So let's also share some evidence for sexual selection. And while evidence for sexual selection, well, you can say, wow, prof, just having sexual dimorphism is evidence that there's something called sexual selection. Why does the male have to look different to the female? Well, you're right. It's evidence as insofar as the fact that all of us are sitting here talking and alive today is evidence that evolution happened. But some people actually need to see numbers. And so real evidence, in other words, from genes that are involved in sexual selection, only actually, believe it or not, it only came up in 2014. The first time genomic evidence underpinning sexual selection actually became known, was discovered, was in 2014. And that happened in a PhD study by this chap here named Poelstra. You see where my mouse is? He is the first author, all of these people are authors, he's the first author of a paper in a journal called Science, and it's one of the Science, as you might or might not know, but you will know it and you will not forget it, is one of the top scientific journals in the world. And so the paper is called The Genomic Landscape Underlying Phenotypic Integrity in the Face of Gene Flow in Crows. That's a bit of a mouthful, but what it is actually saying, and that is why it was published in Science, is that we are providing the very first hard evidence for Mr. Darwin's idea of sexual selection. And this is why this is a fantastic paper, and this is why we are even talking about it. Okay? So, and always the first author is the person who studied or who carried out the study. In this case, this Poelstra was a PhD student, and the last author is always the one who is the professor, right? So Joachim Wolff is the professor. Okay, so Poelstra was a student of Wolff in the lab of Wolff, and all these other people were also in the lab of Wolff, and they all helped Poelstra to achieve his PhD. And his PhD resulted in this absolutely amazing paper in the journal Science. So there are two kinds of crows. In fact, this one looks very similar to a crow, the pied crow we have in South Africa. But in Europe, these are European crows. In Europe, there are two kinds of crows. There's this one which is completely black, okay? And then there's this other one here, which is black on the top of its head, black wings, maybe a bit of black here in its chest, but the rest is what? It's gray, okay? So the torso is gray, the head is black, and the head is, and in this one, everything is black. All right. So what is about these two crows? What is interesting about these two crows? Firstly, they are two different species, and on top of that, what? They look phenotypically very different. This one has a particular phenotype, and this one has a particular phenotype. Right. So let's move forward. There are these two species, okay? And here is a map of Europe. And you see this black line here? This black line tells you where the two species are separated, right? So in Spain and Germany, you have the black one here, this guy here, and on the other side, onto the right of this, or to the east of this line here, in Poland, Sweden, and so on, all of this part of Europe, you have this kind of crow here, okay? And when the two species come into contact, they form a hybrid zone, okay? And this hybrid zone is this black line here, this black line, this curvy black line here, okay? That's the hybrid zone, where these two species come into contact and mix and produce individuals who don't look like this and don't look like this. They look in between, okay? And those in between individuals only are found here in this black zone, this hybrid zone here, okay? Right. The hybrid zone is very narrow. It's very narrow. You see, it's not very broad. It's a thin black strip. Okay. And I ask here, why is a hybrid zone very narrow? And the answer to that question is going to come at the end of the lecture. Okay. So we will answer why the hybrid zone is very narrow. Why isn't the hybrid zone very broad? Like, why isn't it going like from here to here in a thick band across Europe where the two meet? Why is it not a thick band where you get hybrid phenotypes? The hybrid zone is where you get hybrid phenotypes, the hybrid traits. What are the hybrid traits? Are they this? No. Are they this? No. They are in between. They are hybrid. This is one species, this is another. They come into contact here in this zone, and in this zone you get individuals that are halfway more or less in between these two. Okay? They are hybrids. So, let us proceed with the interesting stuff. That was the background. Now, if you look at the genomes of these two species, you take the samples, you look in the black dots, dots here, they're sampled in Germany, they're sampled offshore here on some islands, they're sampled in other islands. So you take samples of the black kind, and you take samples of the black and white kind of species, right? And you do the genetics. And as you know, microsatellites are loci throughout the genome that you can amplify by PCR and figure out whether populations are closely related or not, or whether there's gene flow between the populations and so on. So microsatellites between these populations here showed, and this is really important, it showed that there is no differentiation between the two species. You need to hear that again. These two, the black one and the black and white one, they're different species. They looked at microsatellites in these guys and microsatellites in these guys. And the microsatellites were unable to tell them apart. In other words, the microsatellite said, Icona, it's one species. That's what the microsatellite said. The microsatellite said, there is no differentiation between these two species. Icona, there is one species. Okay? That's what microsatellites told these guys. But you scratch your head and you look at those two and you say, but they are different. Right? They're not the same. They're different. On one side of Europe, you get the black one. On the other side of Europe, you get the black and white one. They are different. How can the microsatellites say that they are the same? How can their genomes say that they're the same? So what did these guys do? They looked, they said, no, we're going to get genomes now. We are done with microsatellites. We're going to take whole genomes. We're going to do bioinformatics. We're going to use our pipelines, all those things I described to you in the first couple of lectures, pipelines and so on. That's what they did here. They took the genome of the black one, the full genome now, not a few microsatellites, the full genome. They took the black one, and then they took the genome of the black and white one. And what did they do? They lined the two genomes up together. They lined them up perfectly together. So every base lined up perfectly with the other base on the other genome. Okay? So they look at the genome of the black and the genome of the black and white. They put them together and lined them up perfectly. So once they lined them up, what could they do? They could start by looking at a tiny piece of one end of the genome and working out in that little piece how much differentiation there was between the two genomes. And then they do that in the next tiny piece of the genome, and the next tiny piece, and the next tiny piece, until they cover the whole genome. And what do they do each time? They calculate differentiation. Okay? Differentiation. And the way they did that is they calculated, and you can see it here on this axis. So this graph is showing you the whole genomes of the two species together, and it's telling you along the genome where are the places where the differentiation was the highest. In other words, where along the genome of the two crows or the two species did you see big differentiation? Because remember, what did microsatellites say? Microsatellites says these two are not even two species. They are the same. No differentiation. Okay? Whereas this plot here is a genomic plot of differentiation. This dashed line here is what? This dashed line, forget about this. This is the X chromosome, so it has a higher level of differentiation. But here are all the other chromosomes, the autosomes. And this dashed black line is telling you that is the average differentiation across the genomes. So it's telling you the average differentiation across the genomes is actually quite low. So it's telling you the same as the microsatellites, right? It's telling you that there's not really much differentiation between these two species. But the beauty of whole genomes comes now, because it's not only telling you that what microsatellites have told you, it's telling you a little bit more information. What more information? Look at this and tell me what more information are the genomes telling that microsatellites could not tell. Okay? All microsatellites could tell was, look, there's this dashed line here. Look, there's low variation. It's low. That's all microsatellites were able to tell. Right? Genomes can tell you a lot more. So what are the genomes telling you? The genome is telling you that, yes, although 99% of the genome shows no differentiation, same as microsatellites, 1% of the genome shows very high differentiation between species. So what is that telling us, guys? That is telling us that, look here, along this, all the low differentiation is below this dashed line. Anything above the dashed line, differentiation values, and in this kind, they measure differentiation by doing an FST between the two genomes, calculating FST. We're going to get to FST later in the module. But just if you're worried, how did they calculate the differentiation? It's by calculating FST between the two genomes along a sliding window across the genomes. So look at some of these places where the FST is high, the differentiation is high. There, here, 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 here. Why? Because it's above this dashed line here, okay? It's up here in this space here. And look at these places where it's really high. It's going up to 0.7, 0.8, yeah? So it's really high in certain parts of the genome. But if you add all these parts with high differentiation, it comes only to 1% of the genome. So it's high, but in a tiny piece of the genome, it's high. And it's low everywhere else. Now, this is becoming exciting. Yeah? This is becoming exciting. Because in that 1%, these guys discovered that that 1% where the two crows, the two genomes were very high, had high differentiation, these guys discovered, well, let's zoom in to those places with the high differentiation. And we will see what genes are there in that part of the genome. In other words, what genes are causing black color and what to be differentiated from the black and white phenotype. Okay? So if you look in the parts of the genome with the highest differentiation, you will find the genes that are actually making these two different species. And it turns out in these areas of high differentiation, here is again mean FST, and you see here it's low, and it suddenly goes up here, and suddenly goes up, and then goes low and low and low again. These areas where it's high, those areas are where this genome and this genome have the most differences. Okay? So look at this here. This gene here, okay? These genes RGS9, PRCKA, CACNG4. These are the names of actual genes that are physically at that point in the genome in these birds. And it's telling us that, well, it's suggesting to us that these genes with the highest differentiation are the ones that are involved in keeping these two phenotypes. Okay? What the title of the paper said, maintaining the integrity of both phenotypes. So basically making sure, even though there's a lot of gene flow, there is no differentiation for most of the genome, but certain small parts of the genome are responsible for making the two species look different. Okay? And those certain parts are the parts you will discover only when you line the two genomes up and you see the places where they are most different, the highest differentiation. And they call these, Poelstra et al., call these speciation islands. Okay? Basically, let's go back to the last slide. Here are these speciation islands. They are basically islands in a sea of low differentiation. So the whole genome is a sea. The two genomes are a sea, and they have low differentiation. And at certain place, you have islands poking up above the sea with high differentiation. That's why they call them islands. Okay? So they call these things, these areas here where you have a spike in FST. Here. These areas here, they're calling them islands because they have high FST between the two genomes. Okay? And they discovered these islands have the genes that make the different colors. So that makes the two phenotypes, the black phenotype and the black and the white phenotype. And these islands also have genes for visual perception. That means when you see something, there's a gene that is determining whether you like what you see or whether you don't like what you see. And there's not one gene, there's many genes that do that preference, okay? But in these two birds, these Poelstra et al., they found out that the genes that are involved in the choice, whether you like something or whether you don't like something, they're also inside these speciation islands. They're also inside these places where the differentiation between the two genomes is very high. Okay? So so the whole genomes, the two genomes, are almost identical to each other. Just like microsatellites said, there's no differentiation. Only when you look at these tiny islands that have very high differentiation do you find the genes in there that are controlling for the phenotype, and the genes that are controlling for the preference of the phenotype. Not just the phenotype, but when you look at the phenotype, which one do you prefer? Those genes are also there in those speciation islands. So if you inherit an island, you basically inherit the phenotype and the preference of one of these species, not both. Are you still with us? If you're not, rewind and listen to this again, because there's no sense in repeating when you have a video, right? You can just go back and listen again. So you can say, okay, Prof, the clever ones among you, maybe you're even saying this. You can say, okay, Prof, just because the two genomes of this, the genome of this crow and the genome of this crow is highly differentiated in these islands, and these islands have these genes. But Prof, just because they're highly differentiated, you don't know that these are the very genes that are causing the color. Do you? Do you know that this gene is causing the black color? Or do you just think it's causing the black color? So this is something that's a very important question. If some of you have asked this question, you have a future in science. That's a very good question to ask. How do you know that these genes in these islands of speciation, these highly differentiated genes, how do you know that they are the real ones causing the evolution, the phenotypes here, the two phenotypes? Well, there's a way to prove it, right? And you do that through RNA-Seq. So what did these guys do? How do we know if the genes on the Speciation Islands are really involved in phenotype? Well, that's where RNA-Seq comes into its own, okay? And that's another reason I wanted to talk about this paper with you guys, so that I can show you in a real example how RNA-Seq proved Darwin's idea, okay? Genomics and bioinformatics is the only way of the future, ladies and gentlemen, okay? That is why if you do genetics with me at Univen, you're only going to be doing genomics. You're not going to see microsatellites. That was 20 years ago. Now it's all genomics. So RNA-Seq, you know what RNA-Seq is because we've been through it in second year. We briefly went through it this year. It's briefly in your course guides. But RNA-Seq is basically extracting all the RNA in a tissue, transcribing it to DNA, and then sequencing all that DNA using NGS. So every messenger RNA that is being transcribed gets sequenced. So you have basically the whole transcriptome, as we call it. It's not the genome, it's the DNA, it's the sequence of all the transcripts. So it's called the transcriptome, okay? And that's what RNA-Seq gives you. It shows you what is actually being transcribed, okay? And so if we look at the expression of different genes, okay? We look at the expression of different genes. So now look at the gene for melanogenesis, which would be this gene here, PRCKA, okay? But there are other genes for melanogenesis. Melanogenesis is what? The black color, right? So let's just look at that one gene. So if we take our sample, two samples from each bird, take a skin sample from the torso, take a skin sample from the head, right? What expression profile are you expecting the head and torso to give when you're looking at the gene for color? It should say what? It should say what? It should give the same profile, right? In this black color, because the head is black and the torso is black, so you should see the same expression of genes in both head and torso. Okay? Right. But what should you see here? If I take, in this bird, if I take a sample from the head and a sample from the torso, here, this white part, I should see the black gene only being expressed in the head sample. I should not see it being expressed in the torso. Okay? Because the torso is not black. So that's a good test. Let's have a look what they found. In the gonads, which are the sexual organs, between the two kinds of birds, right, there were in fact no differences. The two kinds of genes. All the genes and melanocyte genes involved in the black color. In the gonads, the transcription of these genes was the same. All the genes and the melanogenesis genes were transcribed at the same rate. Okay? Because you expect that because it's a liver, right? Those genes are usually not turned on in the liver. The liver doesn't have to be a certain color for phenotype, right? What about gonads? Same with liver. Skin on head, let's look at this. Skin on head, no difference between normal genes and other genes. Okay? Skin on head. But skin on torso, look at this. It's highly expressed compared to all other genes. So in the torso, right, you are getting much higher genes for melanogenesis compared to other genes. And what they showed is that in the head, the same gene as in the head and torso gets expressed, but only in the head for this one. Okay? So if you take a head sample from this species here, it has the same profile as the head and the torso from this species. But if you take a torso sample from this bird, it's not the same as the head, and it's not the same as the head in that one, and it's not the same. Why? Because this one is not producing any black, it's producing only the white color, right? And so RNA-Seq can tell you how much of the black color is being produced in the different tissues, and that way you can prove that prove that the genes that are on the Speciation Islands are in fact the ones that are being expressed at different levels in the different tissues. And that is how RNA-Seq can be used to prove that the genes on these Speciation Islands are the very ones involved in phenotype. So it's the Speciation Island that is controlling the phenotype. That's an incredible discovery. And on top of that, now let's look at, remember when I said we get to the end, why is the hybrid zone narrow? We're going to get to it now. So what happens in the hybrid zone? In this narrow black belt here that you see, where you see my mouse, what happens there? Okay? So there are two kinds of hybrids that are possible. There's a hybrid that looks halfway between these two. Okay? So it doesn't look like this one. It doesn't look like this one. It looks like what? It looks like something halfway in between. That happens when what? There is a recombination event inside the Speciation Island. Okay? So basically, you have a black crow and you have a black and white crow meeting in the hybrid zone and having a baby. Okay? If they do that, the hybrid will look, if there's a recombination event inside the Speciation Island, then the hybrid will look, because then if you have a recombination event, half the Speciation Island goes to one and the other half goes to the other. So you don't get the full phenotype, right? You get a hybrid phenotype. You look somewhere in between those two. But the other kind is when there is no recombination inside the Speciation Island. In that case, that Speciation Island is inherited as is, unchanged by the offspring. Okay? So we can either get the Speciation Island that makes it a completely black crow, or it can get the Speciation Island from the other parent intact that makes it the black and white species phenotype. Okay? So that in that second kind of recombination, okay, the hybrid doesn't look in between. The hybrid looks exactly like either one parent or the other, depending on which island it got intact from which parent. If it got the island intact from this parent, the black parent, it will also look black. If it got the island intact from this parent, then it would also look black and white, exactly like this. But if it's in the first category, where half the island, there's a crossover in the island, inside that Speciation Island, that's high area of differentiation, there's a crossover event during recombination in meiosis. That is why you had to understand what is meiosis and recombination in second year. Otherwise, you are lost right now. But that's not really my problem. It's your problem if you're lost. Okay? So, you have a recombination event inside that high differentiated Speciation Island. And what happens? Half the island goes to one gamete and half the other island goes to another gamete. So any gamete only has half the island. It's not going to look like a black, like one species or the other. It's not going to look like either species. It's going to look like a in between. Okay? And so that's why all of these genes are clustered onto these differentiation islands or speciation islands, right? Because nature wants to maintain the phenotype. And so if it's clustered into a tiny piece of the genome, the chance that you will have a crossover is very small. Okay? So the chance of actually getting a hybrid phenotype gets reduced thanks to this speciation island mechanism. Okay, so since the genes that are for mate preference, so the choice, and the genes for the phenotype itself are both on the speciation island, when there's a recombination event inside a speciation island, the hybrid females will choose hybrid male with mixed phenotypes. But when there is no recombination inside the speciation island, when these two species mate with each other, there's no recombination in the speciation island. There's recombination, but it's not in that spot. Then the hybrid female will choose either one or the other parental phenotypes, depending on what island she obtained, right? So basically, when these two mate with each other and there's no recombination inside the speciation island, the female child from that mating will look like either black or black and white. It will not look like a hybrid. It will look like one of those parents. And if it looks like one of the parents, it will also choose a mate that looks like that parent. Because why? The genes for preference and the genes for color were on the same part of the genome, in the same speciation island. So if there's no recombination in that speciation island, what do you get? You get the phenotype of one or the other, and you get the preference of one or the other. So basically, non-hybrid females always choose the phenotype of their own species, whereas a hybrid female will choose the phenotype of one or the other if the speciation island is inherited intact without recombination. But if the speciation island is inherited in two pieces, or you get only half of it from one and half from the other, then you are not going to have the proper phenotype. And you are not going to be chosen except by who? Another bird who has the very same recombination event. Okay? Another one with a hybrid phenotype. Okay? Which is why, okay, because there's now basically heavy sexual selection against hybrids. If you're a female and you have one particular speciation island, you will choose a male who looks the same. So in other words, even though you are a hybrid, you don't look like a hybrid, and you don't choose a hybrid phenotype. And that is how sexual selection, let me just turn that off. That is how sexual selection is actually selecting against the hybrids. So sexual selection, because the female is only choosing based on the phenotype of one or the other, she is maintaining these two species phenotypes. Despite the fact that they're able to mate, have a lot of gene flow, and the genomes of both species are actually pretty much the same. Despite that, sexual selection is able to keep the two species phenotypically apart because the females choose in a certain way. This is a really, really a brilliant study. And why do I say that? Because it shows that Darwin was right about sexual selection. After more than 200 years after he came up with his idea, in the era of genomics and bioinformatics, we provide the first hard evidence that Darwin was right, that sexual selection is indeed a powerful force that could lead to the evolution of species. Okay, that is the end of our lectures on selection. We are now on and Darwin and his ideas. Now we're focusing for the rest of this section on Mendel and his ideas about inheritance. And you will find me in the next lecture.